Welcome to the next talk in the Bioinformatics CRO webinar mini-series. At the Bioinformatics CRO, we help life science teams turn complex data into clear decision-ready insights, providing flexible expert bioinformatics support from study design through analysis and reporting. As part of that mission, our webinar series features practitioner-focused talks with concrete takeaways you can put to work right away. Today's talk is by James Opzoomer. James is a senior scientist in the Innovation Lab at Relation, where he develops single-cell and spatial genomics platforms to accelerate drug discovery. His projects span high-throughput multimodal single-cell sequencing and spatial transcriptomics technology development, generating ML-ready datasets that power novel therapeutic insights. Today, James will be presenting on biophysics-informed spatial transcriptomics approaches to identify cytokines causally driving downstream gene programs. After the talk, we'll host a live Q&A session. This is streaming both to YouTube and LinkedIn, and on either platform, you can put your questions in the chat or the comments at any point during the talk, and we'll bring them into our discussion afterwards. James, over to you.
Thank you, and hello. So I'm delighted to be speaking today at this BCRO webinar, and I'd like to thank Grant and the BCRO team for inviting me to speak with you today about Relation and some of our spatial transcriptomics work within the innovation team. So I'm going to start today by giving you an overview of Relation and our approach to data generation, and then I'll dive into a novel spatial transcriptomics analysis method that we're developing called BISTR, and provide a worked example at the end. So first, I'd like to start with a question. What are some of the main challenges with the current model of drug development? And why is now a uniquely good moment to deploy large-scale patient genomics to solve this problem? So shown here are four major trends that define the future of drug development and healthcare. And on the left, we have sort of two negative trends. First, that the cost of drugs is ever increasing. We spend more money on healthcare, but we don't see commensurate increases in life expectancy. And this is also demonstrated by the ratio of healthcare spend to life expectancy on the left. Now, the two good trends on the right are that the cost of sequencing is drastically decreasing. You can now do a whole genome sequencing for about $100, and the cost of compute that's driven by titans like NVIDIA has made it more accessible than ever before. So really the problem that Relation is trying to deal with is the first one, decreasing the cost of drugs. And what we want to ask is, can we use these two trends on the right to solve those on the left? Now, this slide really represents a simplified overview of the drug development funnel, which I'm sure you're all well aware of. On the left, we start with maybe 20 programs, 20 ideas for new medicines. And we invest on the order of $1 to $3 billion across this funnel. And after all that work, we typically end up with just one marketed drug. So most of the attrition here is because we were wrong about the biology. So although every stage in the funnel is important, the decisions we make right at the beginning in target discovery echo all the way through this funnel to the clinic, where failure is acutely expensive. And so that's why we believe that target discovery is really the most important problem in drug discovery. So at Relation, our ambition is to transform target discovery into an engineering discipline. And now this means building systematic, repeatable processes powered by large-scale patient data and ML models. So the funnel that I previously showed you is another representation of this statistic on the top left, that over 90% of drugs that enter clinical trials ultimately fail. So how do we transform R&D so that this number looks very different in the future? Now, over the last few years, several large analyses have given us an important clue. So on the right, there are two examples of these. The first is a recent Nature paper where it looked across many clinical programs and the papers from Matt Nelson's group. They show that when a drug target is supported by human genetic evidence, the probability of success in the clinic is increased compared to targets without that evidence. In other words, genetics gives us causal anchors in human biology. The second work shows that single-cell RNA sequencing of human tissue sharpens that picture. So by knowing which cells in which tissues express a genetically supported target, we can better predict efficacy. So how do these approaches fall into historical data collection strategies? So on the left, we have large N, low value, high dimensional observational data. These are things like the Human Cell Atlas, large biobank cohorts. There's a lot of it, but it's noisy, confounded, and often only weakly connected to clear interventions that we want to make in drug discovery. And on the right, we have small n, high value, but low dimensional interventional data, mechanistic experiments in model systems, but in small numbers and with low dimensional readouts, a few readouts and few perturbations. Now, what we actually need for AI-driven target discovery is bespoke multimodal perturbation data that links interventions to rich molecular and cellular readouts across diverse biological systems that are related to primary patient material. Now, that missing data layer is what enables us to train models that actually learn the consequences of perturbing a target in a specific cell type and tissue. And overall, we believe that current models and data in the public domain are nowhere near sufficient to deliver a meaningful impact in target discovery. So we therefore have to build the right data and the right models applied to where they most make sense. So now that I've talked about why we care so much about genetics, single-cell data, I wanted to give a quick overview of how Relation is actually set up to do this in practice. And this slide represents a high-level map of our platform. On the left, you see human tissue profiling. This is where we generate deep multimodal data directly from patient samples, whole genome sequencing, single-cell spatial transcriptomics, single-cell transcriptomics, and proteomics. Now, all of this is connected to the cellular modeling teams who run perturbation experiments on patient-derived primary cell systems to generate bespoke data for the models. And this connects to translation pharmacology who take the prioritized drug targets and turn them into drug discovery programs. Now, this is all connected to both data science and our three main machine learning platforms. Rosalind, which identifies genetically validated drug targets, Ada, which focuses on reversibility, and Turing, which provides drug discovery context of our targets. And I'm not going to go into these platforms in detail today because I really want to focus on the spatial genomics data that we generate in human tissue profiling and some of the new analysis methods that we're developing to better use our spatial transcriptomics data in drug discovery. So as an example of the type of primary patient data that we collect, I just wanted to show a case study of osteomics. This is our flagship observational clinical study focused on osteoporosis and bone disease. So in this study, we partner with orthopedic surgeons across London to collect human bone waste from key surgeries. So these are total joint replacements, elective surgeries associated with osteoarthritis, and hemiarthroplasty. So these are non-elective surgeries resulting from osteoporotic fracture, really the end stage of osteoporosis. So from each patient, we build a genuinely multimodal data set. So that's whole genome sequencing to identify variants in genes that causally influence bone density, fracture risk, and response to therapy. And this feeds into our genetic discovery platform, Rosalind. We also generate single nucleus RNA-seq of bone and joint tissue to map those genetically supported targets into specific bone, stromal, and immune cell types and states within the tissue. And this sharpens our view of where these targets are expressed within the tissue. We also collect blood-based proteomics to find circulating biomarkers that report on pathway activity can be later used for patient stratification. And in addition to this, also rich clinical metadata including BMD or bone mineral density, to anchor everything back to quantitative phenotypes. And now this lets our models learn how genetics and cell state translate into real clinical outcomes. So in addition to the single cell RNA-seq, we generate spatial transcriptomics data with Xenium and the Visium HD platforms on human bone and other tissues associated with the other disease programs we're working on. And this is really important because single cell data tells us what cell types and states are present within the tissue. But really, we lose where they sit in the tissue and how they interact and communicate with other cells within this spatial context. So together, these genomics and single-cell datasets give us a dense, patient-centric view of disease biology. And in particular, we in the Innovation Lab are interested how we can utilize this spatial transcriptomics data to disentangle the causal microenvironmental signals, so the cell communication pathways that drive cell state and cellular response to microenvironment. And this has led us to develop a new analysis method called BISTR, or Biophysics-Informed Spatial Transcriptomics Regression, that I'd like to share with you today. So spatial technologies are key for preserving the in situ cellular context present in tissues, providing a contextual perturbation system of sorts to understand some of the microenvironmental signaling factors that may be driving a particular cell state within a tissue or within a particular disease. So we're often attempting to model our disease states in less complex in vitro systems, like some of the ones shown here, 2D cell models and 3D organoids or organ-on-chip models. And the kind of the motivating feature of this BISTR package is to answer some of these questions. It's can we identify cytokines responsible for cell identity and behavior in primary patient tissue? And could we then stimulate cell models to mimic some of these disease-relevant or patient-relevant microenvironmental niches? And we hope that this can add value to the drug discovery process and to kind of our efforts in vitro cellular modeling by using this knowledge to build experimental systems with greater disease relevance in vitro. So a lot of this work is enabled by the advancements in the resolution of spatial genomics technologies, which is really rapidly changing. And we recently published a review in Cell Genomics tracking these technology trends called SC trends. This kind of summarizes the historical development in spatial omics technologies, as well as some of the analysis packages available. And we also comment on these kind of developing spatial technologies in real time, since it's such a fast-moving field, at our blog sctrends.org. So I encourage you to check it out if you're able to. So the work that I'm going to show you today is really focused on 10x Genomics' Visium HD platform. And this is one of these spatial sequencing-based spatial transcriptomic technologies where the increased resolution in this generation of platform, now 2 micrometers, has really enabled subcellular resolution, allowing us to track several biophysical processes that are shown out here on the right. So RNA abundance, RNA localization, and also RNA splicing at the subcellular level. And we can use these 2 micron pixels to approximately reassemble single cell data based on image segmentation tools in the imaging modality to create approximately single cell data. So this slide sort of positions BISTR among other spatial modeling approaches. On the left are sort of simple heuristic-based approaches, like using a radius around a specific cell or a KNN neighborhood and computing sort of some summary statistics. They're fast, but the spatial scale is often somewhat arbitrary, and the tissue is treated more like a discrete bin than a sort of a continuous space that it is. On the right, we've got deep learning-based approaches. Now, these can be powerful, especially when they leverage analysis pipelines from the image space, or are often paired with single-cell data, but they're typically more data-hungry and sometimes less mechanistically interpretable. So BISTR sits in the biophysical model space in between. So we encode this process of intracellular signaling via ligand diffusion as a diffusion decay problem with boundary exchange to generate interpretable per cell exposure features without choosing an ad hoc neighborhood. It runs with more modest compute and also sets up a clean entry point for ML once the inverse problem is well posed. So this is sort of a schematic representation of the BISTR computational pipeline. You have your underlying biological system, and you generate subcellular spatial transcriptomics data, say 10x Visium HD data. You then use an image segmentation vision transformer, for instance, to identify nuclei and cell boundaries and infer subcellular compartments. You then quantify the transcripts on the nuclear and cellular level. And then we construct the extracellular domains, the space between the cells, as a finite element triangulation mesh. And we model paracrine signaling fields per ligand across this mesh using a finite element methods. This allows us to extract the per cell signaling features, which we identify with receptor gating, so understanding the concentration of the ligand at a cell boundary and whether the cell expresses the cognate receptor to this ligand. And from that, we can characterize which ligands predict certain gene expression via a GLM-based model. So this is another schematic that represents the data flow within the BISTR Python package. You have your Visium HD data. You identify nuclei with a vision transformer, and you perform morphological expansion of cells to create like a cell cytoplasm boundary, giving you approximately single-cell data. You then build the FEM triangulation network. You use public databases to look up ligand and receptor genes that are expressed within your cell types of interest, and you solve the FEM network across all of your ligands within the intracellular space. Now, this gives you the FE solution at the cell boundary, and we also look at ligand flux, which is the relationship between the expression of a ligand within the cell and the FE solution at the cell boundary, effectively identifying whether a cell is a source of a particular intercellular communication ligand or a sink. Is it just receiving this signal? And then we use a GLM to identify which ligands are most predictive of certain gene expression programs downstream. So now I want to show you kind of a worked example on a publicly available Visium HD dataset. So this is the BISTR package applied to this 10x genomics colorectal cancer dataset. This is a 10x Visium HD FFPE dataset that was published as part of the preprint that was released along with the Visium HD product launch in 2024. So here you can see a high-level view of the H&E image of the tissue that has been assayed, and zooming in onto a smaller subsection of the tissue so you can see the individual cells. We use a vision transformer model to perform nuclei segmentation and then morphological nuclei expansion. So we follow this expansion to assemble the 2 micron spots into approximately single cell data, which we annotate with its various cell types, giving us a tissue representation of single cell data that looks like this. Here they're colored by their cell type annotation. So on the left here, you can see we construct the extracellular domain and mesh. So we triangulate the extracellular space between the cells whilst using a tissue mask to limit the extracellular triangulation to the space that's only underneath tissue. And starting with a ligand expression per cell, we formulate an FEM problem with diffusion and decay parameters plus Robin membrane coupling that allows us to solve a sparse linear system per ligand and get the FE solution across the tissue space. And here you can see the cells themselves are colored by the expression of ligand VEGF-A, and you can see the FE solution in the intracellular space colored in this sort of white to red heat, showing that cells expressing high VEGF-A are predicted to secrete VEGF-A into the intracellular tissue space, and we model this diffusion with decay throughout the tissue. And this ultimately gives us a FE solution across each of the communicating cells within the tissue, which we gate, basically binarizing them based on whether they express the receptor to a particular ligand or not. If they do express the ligand, then we calculate the FE solution across the cell membrane of each cell and also the flux. So this is the average membrane exchange signal. So effectively, this is the proportion of the ligand expression within the cell and at the boundary of the cell from the extracellular space. Is this cell a source or a sink of this intercellular communication signal? So in order to understand what ligands might be affecting certain cell types, we found that the coefficient of variation and also, in a related sense, looking at the mean ligand flux versus the standard deviation of the ligand flux is informative to understand the kind of most variable intercellular communication ligands across a tissue and cell type. So in this respect, in this particular example, we're looking at VEGF-A here in tumor cells, which has a relatively high mean flux across this tissue section. So we use then a negative binomial GLM fit to the per cell gene counts, which has predictors such as receptor-gated ligand exposure. So the coefficients of this model quantify how exposure shifts expected expression. And we can see which exposure to which ligands are related to specific genes and then gene programs. Here we can see that our model captures the directionality of many genes known to be associated with VEGF-A exposure in tumor cells. And this indicates that we're capturing known biological processes associated with this ligand sort of ligand receptor interaction in this tissue. So in closing remarks, I think we often find that sequencing-based spatial transcriptomics technologies have a lower UMI coverage that's somewhat sparser than single-cell RNA-seq. This has motivated us to develop novel tools to understand the relationship between intracellular ligand receptor signaling and downstream gene expression. So this tool that we developed, BISTR, converts spatial transcriptomics counts and in coordination with segmentation into physically constrained extracellular ligand fields, and then into per cell exposure for downstream modeling of the effect of ligand exposure on gene expression. And we believe that modeling ligand receptor interactions like this with a biophysics constraints gives more interpretability into the intercellular signaling process. And we've designed this BISTR method as a flexible toolbox that is deployed as a Python package, which we hope to make publicly available sometime soon. The goal of this approach really is to generate more tissue contextual, experimentally testable hypotheses, especially where simple in vitro systems miss microenvironmental signaling context. So we can better understand the intercellular signaling processes that drive cell states in patient tissue, and in particular to better understand disease. So we will be publishing this approach hopefully as a preprint soon, and so I encourage you to keep your eyes out for it at that time. So yeah, thank you for listening today.
James, thank you very much. So does the BISTR package work with spatial transcriptomic technologies other than Visium HD?
Yeah, so it's designed from the core methods within it, designs within a spatial sort of transcriptomics method agnostic approach. Really, I hope that I kind of highlighted that what you need is subcellular resolution spatial transcriptomics data, and from there you can reassemble sort of approximately single cell and whatever compartment you can segment with your sort of image layer into that form of data. So Visium HD is great for that, but we're excited to get our hands on hopefully the new Illumina spatial transcriptomics technology that's coming out that appears to be sort of in this 1 micron resolution. But yeah, it should work across different spatial transcriptomics technologies, although we have only tested it with Visium HD, but we hope to expand that outwards soon.
Thanks. Now, what makes the BISTR package biophysics informed rather than just a spatial regression?
So that's a good question. So the kind of BISTR approach explicitly models paracrine signaling as a spatial field within the extracellular space using this diffusion with decay FEM approach solved over effectively the finite element mesh that we build from the native tissue geometry from the spatial transcriptomics, the sort of the imaging data and the spatial transcriptomics data itself. So this kind of, we believe this builds a more representative intracellular communication space than just representing cells as nodes on a graph without understanding, the distance, but also some of the tissue-specific features that might exist within it. For instance, a future direction that we hope to go is to use image segmentation within tissues to create different tissue zones, right, which you can identify from H&E and other types of immunofluorescent staining, where ligands might have difficulty passing through. I'm thinking in particular, we work a lot on bone, as I touched on, but using that to create more representative data.
Great. Well, thank you, James, and thanks to everyone for joining us. Join us for our next webinar on February 18th at 11 a.m. Eastern. Phil Ewells from Sakara will discuss reproducible bioinformatics at scale, NF-Core, and Nextflow. Thanks.
Thank you.